Hello, everyone, and welcome to today's Keynote webinar titled Ultra Rapid Nanopore Whole Genome Sequencing in a Critical Care Setting. This webinar is a part of the 10th Annual Genetics Virtual Week. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the Presentation window and follow the process to obtain your credits. I now present today's speaker, John E. Gorzynski, DVM, PhD, scientist, Stanford University. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dr. Gorzynski, you may now begin your presentation.
Thank you so much for that introduction. I am John Gorzynski. I am a scientist and postdoctoral fellow at Stanford University in you and Ashley's lab. And today I'm going to give you a brief overview of the history of whole genome sequencing. I'm going to show you how advancements in these sequencing technologies has enabled us in the you and Ashley lab to develop an ultra rapid genetic test for critically ill patients. Which is an accomplishment that has been awarded a Guinness World Record for the fastest genome sequencing technology. But more importantly, it enables us to diagnose patients at a rate that was something we could only dream of a few years ago. And being able to make a rapid diagnosis, we're able to have our patient's healthcare team provide the best care possible. So in order to demonstrate the clinical utility of this ultra-rapid test, at the end of my talk, I'll go through a clinical case of an adolescent boy who was suffering from heart disease, and I'll show you how we came to a diagnosis using this diagnostic platform. So to get started, in June of 2000, the Human Genome Project, which was a project that set out to sequence the entire human genome, announced that they had been successful. And this project took a total of 10 years with 10 countries in collaboration. And to sequence the first human genome, it cost multiple billion of dollars. So it's estimated that the cost of the first whole human genome was about $3 billion. And while the Human Genome Project reported at the time that it had sequenced the entire genome, it wasn't really sequenced in its entirety. There were a few hard to sequence regions that were left incomplete. So there were a few holes that were missing within the genome. With that said, they certainly sequenced the majority of the genome. And this was the birth of genome sequencing. Now, in this New York Times article, you can see the man with the glasses on the left. That's Francis Collins. He was the head of the National Institutes of Health until just recently, and he played a huge role in moving this project forward. And standing next to him is Craig Venter. He also played a huge role in pushing the first human genome sequence to be complete. Now, Craig Venter, that guy standing next to Francis Collins, he sequenced his genome second. So he was the second whole human genome to have been sequenced. And at that time, when Craig sequenced himself, it cost a total of $100 million. So previously, I said that the first genome cost $3 billion, and Craig was able to advance the technology enough to reduce that cost by a whole order of magnitude, which was pretty phenomenal. With that said, $100 million is still very expensive, and at the time, we couldn't even think about integrating this into our healthcare system. With that said, though, over the next 10 years, genome sequencing costs reduced so greatly it was compared to Moore's law. Now, Gordon Moore was the co-founder of Intel, the semiconductor company that drove the technology boom in Silicon Valley and is still driving the technology boom today. But from 1970 to around 2000, the technology in Intel was moving so quickly that the number of transistors in an integrated circuit was doubling every 18 to 24 months. And the reduction in cost of genome sequencing was compared to Moore's law because it was reducing so quickly. With that said, when we look at this graph, we can see that between 2001 and 2008, the cost reduction in genome sequencing kept up with Moore's law. However, in 2008, genome sequencing left Moore's law in the dust. Now we can sequence today in 2022 a medical grade human genome for less than $1,000. Now, let's think about that for one more second. The first genome ever sequenced was $3 billion, and now we're down to less than $1,000. This cost reduction is something that we've never seen in the entire world for pretty much anything. But to put that into perspective, say you've been eyeing this brand new Tesla. If it's fully loaded, you're going to be spending about $150,000. And for a scientist like me, that's not affordable. With that said, if we saw the same reduction in cost for this Tesla as we've seen for genome sequencing, this Tesla would cost less than 1 cent today. So essentially, anyone who wanted a Tesla could have it. More importantly, though, the cost reduction of genome sequencing has led to really great scientific accomplishments. One of them, and one of the most recent ones, is that the whole human genome has actually been finished at this point. Remember, in 2000, there were still some sections of the genome that hadn't been sequenced. But earlier in 2021, scientists from the Telomere to Telomere Consortium reported that they had filled in those holes. And this was a result of advancements in sequencing technology, as well as the cost reduction. Also, we've been able to start to integrate genome sequencing into our healthcare system. And this started in around 2010 when the first whole human genome was brought into a clinician's office, and they were able to identify the cause of death for a young man who died of a sudden cardiac death. This was the start of genomic medicine. And rather than interrogating just very small parts of the genome, now that we were able to sequence the whole human genome, we were able to look at the entirety of the genome and identify genetic variants, which may be unique to a patient, and make clinical diagnoses for the clinical presentations that they were going to their doctors with. And with this can allow for more precise medical management. And this was termed precision medicine in around 2010. So it probably comes as no surprise to you, as this is a genetics talk, and we're going to be talking about making genetic diagnosis, that humans suffer from genetic disorders. And it's not uncommon that humans have some form of genetic disorders. As of today, over 6,800 Mendelian disorders have been reported in humans. And of these, I mean, genetic disorders can present later in life, but equally, they can be present at birth. They can be showing their signs immediately after birth or even during the gestation period. That said, 12 to 33% of neonate or pediatric ICU hospitalizations are due to some sort of primary genetic disease. And these hospitalizations where the patient is suffering from a genetic disease is on average 40% longer than those without a genetic disease. We know that we can use genetic testing in order to identify what is the cause of these genetic diseases. And more specifically, when we think about using whole genome sequencing as our diagnostic test, we can have diagnostic rates ranging anywhere between 20 and 69%. And when a genetic diagnosis is made within these patients, there's going to be a whole range of benefits. One, hopefully we'll be able to improve the health of the patient. Two, we should be able to improve the family experience of care. So you can imagine if a mom and a dad have a baby in the hospital, they want to see what's best for their child, right? And if we're able to make a diagnosis and give them the best possible treatment, that's going to improve the family experience of care. Equally, when we think about it on the other hand, the clinician's experience, if they're able to make a diagnosis and get their patient healthy, that's going to improve their experience in the hospital. And on top of that, we're improving experience for the patient, the family, the clinician, but we can also reduce the cost of healthcare. If we make a genetic diagnosis and we can provide the best quality of care to these patients, hopefully we're able to get them out of the hospital quicker. And with that, we can see a reduction in cost for healthcare, both to the insurance providers as well as the out-of-pocket costs for these families. So let's talk about genetic sequencing in terms of making diagnosis. So let's imagine that a baby is admitted to the hospital for some sort of clinical disease. At the time of presentation to their doctor, the doctor is going to perform a physical exam, and with that, they're going to start to acquire data. And with that data, hopefully they'll be able to start to decide what type of diagnostic tests will be performed or can be performed in order to come to a definitive diagnosis. Of these diagnostic tests, genetic testing is becoming more and more available all over the United States. And hopefully with that genetic test, we're able to make a definitive diagnosis and provide precision care to the patient, which hopefully will get them to go home. Now, the problem right now is with genetic testing, specifically whole genome sequencing genetic testing, the standard of care turnaround time for these types of tests is anywhere between 6 to 8 weeks. Now, we can imagine if a patient is in a critical state, waiting 6 to 8 weeks for a diagnosis just isn't feasible for them, right? So other hospital systems have identified this as a problem, and Baylor College of Medicine in Texas has developed a rapid genome sequencing test that has reduced this turnaround time to about 7 days, which is fantastic. Rady's, a children's hospital here in California, has even reduced Baylor's turnaround time to about 3 to 5 days for a whole genome sequencing test turnaround time. But the problem is with these genetic testing and genome sequencing tests, these technologies are using what we call short read sequencing. And we'll talk more about what short read sequencing is and how that it can affect the turnaround time in just a few minutes. But here, at Rady's, they actually set the world's record earlier in 2021 for making a genetic diagnosis using whole genome sequencing at about 14 and a half hours. But we decided here at Stanford, rather than using short read sequencing technologies, we were going to switch it up a little bit and go towards a new sequencing technology called Nanopore sequencing. And we'll talk more about the technology in just a minute. But we decided to throw our hat in the ring and see if we could reduce this turnaround time to less than a day, if not a few hours. So Nanopore sequencing is a way that we can sequence native strands of DNA. So essentially, we can take DNA directly from a patient. We can extract the DNA from cells of a patient, whether it be blood or other types of cells. And from there, we can introduce this DNA into one of these microfluidic devices known as a flow cell. Now, the flow cells, you can't really tell the scale of them right now, but they're about the size of a USB stick, so really quite small. But within this microfluidic device known as a flow cell, there are nanopores embedded onto a biofilm. And when we introduce our native strands of DNA to these nanopores, they can be brought to the nanopore, and the DNA is pulled through. And as the strand of DNA is pulled through that nanopore, there's going to be a change in conductance, electrical conductance, as it's pulled through, which can be associated with the DNA sequence. So remember that DNA is a series of A, T, C's, and G's, more commonly known as bases or nucleotides. And as the strand of DNA is being pulled through that nanopore, the change in electrical conductance will then be translated into this funny graph-like thing that we can see next to the nanopore. We call that a squigglegram. And using machine learning algorithms, we can translate that into the DNA sequence. Now, nanopore sequencing is read length agnostic, meaning any size of DNA that we introduce into this nanopore can be sequenced. Now, short read sequencing technologies, the sequencing technologies that are used by Ray's and Baylor, can only sequence about 150 base pair fragments of DNA, so very short fragments of DNA. And when we think about the entire human genome, which is 6 billion nucleotides or 6 billion base pairs, sequencing at 150 base pair units is very small. And when we think about this in terms of a puzzle, and we think about short reads or short read sequencing, in order to cover the entire 6 billion nucleotide genome of a human, it's going to take a lot of little tiny fragments of DNA in order to sequence the entirety of that genome. We can think about that in terms of a puzzle. So if we had a 1,000-piece puzzle, it's going to take us a very long time in order to identify where each piece of that puzzle goes, similar to that of short read sequencing, when we take the small fragments of DNA and try and align that to a reference genome. Now, clearly, when we look at it in terms of this, the benefit of long read sequencing or sequencing something or fragments of DNA that's longer than 150 base pairs will allow us to put this puzzle together much quicker, right? We can put together a 20-piece puzzle much faster than we could put together a 1,000-piece puzzle. And this is where one of the main benefits of long read sequencing comes in. So, like I said, we decided that we were going to use nanopore sequencing in order to try and reduce this turnaround time for genetic testing even further. So we set our study goals to focus on patient care. We wanted to develop a genome sequencing test that can rapidly and confidently identify clinically actionable genetic variants in order to provide precision care and reduce hospitalization time for our patients. And our two main goals here, like I said, is to reduce genome sequencing turnaround time, but we wanted to do that to be more in line with a clinical decision-making process. So we can imagine if we send out a stat blood test, that it may take 4, 5, or 6 hours to get those results back. We wanted this genetic test to come back in a time that's comparable to that of standard blood tests, so doctors can have all of the information available at a single time to make the most appropriate diagnosis and provide the patient with the best care possible. The second aim or goal of our study was to show that this new sequencing technology developed by Nanopore is a robust sequencing platform that can be integrated into a clinical setting. So, in order to do this, we developed a workflow that consists of four main parts. Um, the wet lab, where we do sample preparation, the sequencing, where we're actually sequencing the entire genome, and then in the dry lab, this is where we are aligning all of those reads to a reference genome. And then we're able to identify variants within the patient by making simple comparisons from the reference genome to the patient's genome. And from there, we can compile a list of all of the variants or genetic mutations that may be associated with the patient's clinical presentation. And then we can send it to our genetic counselors where they can identify which of those genetic variants may be associated with this clinical presentation. From there, doctors can make a final diagnosis. So in terms of making this workflow go extremely fast, we needed to optimize each stage of this workflow. And to get started, we needed to figure out a way that we could extract a lot of DNA from a small amount of blood. So remember, initially, this study was targeting young patients, and you can only extract a certain amount of volume of blood from these patients because they're so small. So we targeted 2 mils of blood, and we wanted to see how much DNA we could get out of a small sample of blood. From there, we optimized and scaled the library preparation in order to get this DNA prepared to be sequenced. Once we were able to extract enough DNA and prepare the library, then we decided that we would distribute this sequencing library, or the DNA that's prepared for sequencing, across 48 flow cells on the Oxford Nanopore Promethean sequencing device. Now, this is what the Promethean sequencing device looks like. We have 6 columns, and within each column on this machine, we can fit 8 flow cells. So a total of 48 flow cells can be loaded onto this machine. And again, we can't really tell the scale of this, but really this is on the size of a desktop tower turned over on its side. So it's really quite small. And we can sequence a lot of DNA in a short period of time. Now, a single flow cell can sequence about 150 gigabases worth of data. So this is about 30 to 40 times coverage of the human genome in 48 hours. But when we take our sequencing library and distribute it over 48 flow cells and sequence all 48 flow cells simultaneously, we can get that same genome coverage within 2 to 3 hours. So this device really enabled us to sequence at an extremely fast rate. So while we're sequencing, once the sequencing library was distributed over the 48 flow cells and we start sequencing, we then had to figure out a way to process this data as quick as possible. So we decided that we were going to upload this data to the cloud, and I'll talk more about why we decided to do that. So if we take a look at the sequencing device here, you can see the Promethean sitting in the middle of this slide, and next to it, there's a computing tower. Now, this computing tower is quite powerful. It has 4 Tesla V100 GPUs, which is the processing component to this computer, and it also has 96 CPUs, which is also another component of the processing. With that said, if we use this computing tower in order to translate the change in electrical conductance to our sequence of DNA, and we do it in a linear fashion, the base calling, or just the part of translating that electrical conductance signal into the ATCs and Gs, that would take a total of 16 hours. And then on top of that, aligning those sequences to the human genome, to the reference human genome, would take another 2.5 hours. So if we sequence for an hour and a half, we're looking at 19, 20, 21 hours in order to get this uh, sequence to the alignment stage. And this isn't even to the point where we've called variants and are able to get that to the genetic counselors and then to the doctors in order to make a diagnosis. So we weren't really pleased about using this computing tower, and we wanted to find a way that we could make this go even faster. So our solution was to upload all of the sequencing data in real time to cloud storage. And on cloud, we could then use the cloud computing services in order to scale up and parallelize all of our base calling and our alignment. And with that, we were able to reduce the time needed from that 20-ish hours down to about 2 hours in total. So in real time, the sequencing data was pushed to the cloud, and base calling was occurring immediately once that data was received on the cloud. And once base calling occurred, those sequences could then be aligned. Immediately after those sequences were aligned to the reference genome, we could then call variants. And again, we did this on the cloud. And we used the state-of-the-art variant calling software called Pepper Margin Deep Variant to identify very small changes in the genome. And we used another program called Sniffles to identify large variants within the genome called structural variants. So these are things like extremely large insertions or deletions of base pairs within that genome. So this is what we did to make the dry lab go fast. And as soon as we were able to have a variant call file or a file with all of the variants identified from a single patient, we then sent that up to our genetic counselors, which developed a decision tree where these variants are prioritized based on a whole range of criteria. And once they were able to prioritize variants, then they could manually go through each one of these variants and identify which one of these is most likely to be causing the clinical presentation of these patients. So I'm not going to get into the specifics of the variant prioritization, but essentially this is what it looked like. We took all of the variants from the patients, and on average, we'd identify about 4 million variants per patient. And remember, this is just a small fraction of the entire genome, which consists of over 6 billion data points, right? So then we go and filter these variants down based on different criteria that would make the likelihood of these variants more likely to cause disease. And as soon as we were able to have that prioritized list, and the genetic counselors could then identify the variants that are most likely to cause clinical disease, we'd send that over to the clinicians. And with that, hopefully they would able to make this a definitive diagnosis and provide the patient with the utmost quality care. So we took this pipeline that we developed. It took us a few months to put the architecture of this pipeline together. But initially, we enrolled a total of 13 patients that ranged anywhere from 2 weeks old to 57 years old. And they ranged in terms of their clinical presentations from having neurologic disease, so refractory seizures or developmental delays or cardiovascular disease. So a wide range of different types of diseases that these patients were experiencing. But the common theme amongst all of them were they were in a critical state. So they were really unwell. Of these 13 patients, we were able to make a diagnosis in 6 of them. With that said, I'm going to present one of these cases. case number 2 to tell you more about how this pipeline works and how patients can benefit from this type of technology. So our second patient, which we named FAST-002, was a 13-year-old male with no history of heart disease. He presented to his pediatrician with a 2-week history of a dry cough when lying down, a decreased appetite, intermittent chest pain and fatigue, and no syncopal episodes had been reported, so no episodes of him passing out. When he presented to his pediatrician at the time, it was thought, hey, maybe this is COVID. So he was tested for COVID, which came back negative. So the pediatrician decided to order a few more tests. The first test was a thoracic X-ray or an X-ray of his chest cavity. And that X-ray identified that this patient had severe cardiomegaly or an enlarged heart. So the next thing the pediatrician decided was to order echocardiography, which is an ultrasonography of the heart. And at the time, the patient was identified to have a left ventricular ejection fraction of 29%. Now, normal ejection fraction or a left ejection fraction is going to be anywhere between 45 and 55%. So this patient having an ejection fraction of 29% was severely compromised. This patient was then transferred to Stanford Hospital and echocardiography was repeated. And at the time, his ejection fraction had reduced to about 21%. So over the course of just over a day, this patient's heart was starting to fail. In addition to a significantly reduced ejection fraction, the left atrium was noted to be dilated and thin, and there was also severe right ventricular dysfunction. And for those of you who may be interested in the imaging of this patient's heart, this is what his echocardiography looked like when he arrived at Stanford. So this is a cross-section of the heart. And when we look at the left image here, the crescent-shaped black patch above is going to be the right ventricle. And then the more circular structure that's sitting in the middle of the image is going to be his left ventricle. And what we can see here is the walls of his ventricle are thin, much thinner than what we'd expect. Also, the image on the right, this is indicating his blood flow, which we were able to determine his ejection fraction, which, as noted previously, was severely reduced. So at the time, it was thought that this patient was most likely experiencing a form of dilated cardiomyopathy. But the type of dilated cardiomyopathy was not clear. It could be due to myocarditis, which is an inflammatory process within the heart, or a genetic cardiomyopathy. And for these types of diseases, it's really important to identify what the cause of the dilated cardiomyopathy is. Because if it's myocarditis, if it's due to myocarditis, oftentimes these patients will get better with medical management. However, if it's genetic cardiomyopathy, these patients will experience rapid decline and oftentimes needs extreme or pretty invasive intervention in order to save them. So we decided to enroll this patient into our study, and this is what our sequencing pipeline looked like. We collected 2 mL of blood, and with that, we extracted the DNA, which took a total of 62 minutes. We then continued to prepare the library, which consisted of DNA fragmentation and the entire library preparation. And in total, this took 4.2 hours. We then distributed that sequencing library across the 48 flow cells on the Nanopore Promethean, and we sequenced at a rate of 1.26 gigabases per minute and generated a total of 204 gigabases worth of data. So this covered this patient's genome 60 times over, in fact, 63 times over, which allowed for us to call variants with high confidence. We sequenced for a total of about 2.5 hours. And with that, during sequencing, like I said, we transmitted all of this data to the cloud, and base calling and alignment and variant calling all occurred on the cloud computing service, which took an additional 3.6 hours. Once we had the variant call files or the compiled list of all of the genetic variants that were identified in this patient, the genetic counselors put it through their decision tree or their variant prioritization schema, and they identified a few variants that they or that were highly prioritized. And with that, they identified a single variant in TNNT2, a gene that we'll talk about in just a second, that was most likely to be causative for this patient's dilated cardiomyopathy. This took a total of 11 hours and 3 minutes to come to that diagnosis of a variant in TNNT2. And when we look at the IGV pileup, or this is a method that geneticists use to look at the genetic variants among many different sequences within the patient's genome. And we can see, although it's a little bit small, within this red box, there are multiple purple boxes, which indicate that there was a slight duplication of 3 base pair sequence, which disrupted this gene. We then did further clinical testing in order to verify that our experimental technique was in fact identifying a variant, and we used a traditional sequencing method called Sanger sequencing. And I won't get into the details of this, but in fact, when we looked at this patient's segment of DNA where we identified the variant in TNNT2, we did find that there was an insertion of this 3 base pair nucleotide sequence that disrupted the gene. We further tested the parents, so the mom and the dad, to see if they had this mutation, to see if this was a variant that was passed from one of the parents, or if this was a variant that was developed while the patient was developing himself. And we found, in fact, that this variant was not present in either one of the parents. So we termed that a de novo variant. Now, more about TNNT2. TNNT2 is a gene known as cardiac troponin, and it's a subunit of the troponin complex, which is an integral structure of the myosin filament, which is a part of the cardiac sarcomere. And the cardiac sarcomere is the most basic contractile unit of the heart. So remember, the heart is squeezing and relaxing all day, every day. And when the cardiac sarcomere or genes within the cardiac sarcomere are becoming disrupted as a result of genetic variation, the function of the heart may not be as good as one would expect. So variants that have been previously identified in TNNT2 have been associated with dilated cardiomyopathy, so a thinning of the ventricular wall, hypertrophic cardiomyopathy, a thickening of the ventricular wall, or restrictive cardiomyopathy, where the heart just isn't squeezing as properly as it should. And this identification of the variants within TNNT2 within our patient fit perfectly with this dilated cardiomyopathy diagnosis. So going back to this patient, the 13-year-old male with no history of heart disease, who had presented to his pediatrician with a dry cough and decreased appetite and some other clinical signs, had genetic testing done, and the genetic testing identified a de novo heterozygous 3 base pair insertion in TNNT2, and it was classified as likely pathogenic. So with that, we were able to make a diagnosis of genetic dilated cardiomyopathy. And the outcome for this patient was the next morning after we made that diagnosis, in just 11 and a half hours, he was placed on the cardiac transplant list. And 21 days later, after hospitalization, the patient received a heart transplant, which was much quicker than anyone could have ever imagined. Now, patient number two was in the first phase of our trial. And you can see it's the second line from the bottom here. And while we reduced the turnaround time from our initial patient, which was hovering right around 19 hours, we took about 7 hours off of that turnaround time. But in between each one of these patients, as we move up, we made implementations to the pipeline to identify what would be the best way to make quick and accurate diagnoses. And once we got through 7 patients, we were pretty confident that we had identified the best method to do that. So we moved on to the second phase of our trial, where we focused on maintaining the turnaround time to as quick as it could be, in hopes that we could keep it less than 8 hours. And you can see that in phase 2, which is the upper part of this graph, that we were able to reduce turnaround time to, on average, 8 hours, with the fastest diagnosis being made in 7 hours and 18 minutes. So this is much faster than the previous world record that was set by Rades. And we were able to standardize this turnaround time to be in line with clinical decision-making projects. But this wasn't all about, making a really fast diagnosis. This was more about providing the best patient care that we could for our patients. And when we look at this graph, we can see that we've greatly reduced the standard of care turnaround time to something that's going to benefit our patients in terms of making diagnoses and hopefully finding the most appropriate type of treatments that we can give them. So with that, we set the world's record at making the quickest genetic diagnosis using the fastest genome sequencing technology. And we've been awarded the Guinness record as mentioned, and as you can see in these photos. Equally, we've been able to publish all of our research in some pretty cool journals, including the New England Journal of Medicine, Nature Biotechnology, and Circulation Precision Genomics, or Precision Medicine and Genomics. And we're really happy with the way that the first part of our trial went, and we're really looking forward to moving forward and identifying new ways that we can make this go even faster. Now, with that said, this was not a one-person job. In fact, this was a huge collaboration between different academic institutions, including Stanford Medicine, University of California, Santa Cruz, Baylor College of Medicine. And we also had a lot of industrial partners as well, including Oxford Nanopore Technology, Google, who use their cloud computing services, as well as NVIDIA, who helped us with all of our hardware. So with that, I'd like to say thank you to everyone for listening, and I'm more than happy to take any questions that you may have.
Thank you, Dr. Gorzynski, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. Our first question is, what are the next steps in this project, and do you have any specific aims for the next phase?
Yeah, thanks. That's a great question. So the next steps in this project are really to scale it to a point where we can do many more patients. So this initial part of the project, we only sequenced 13 patients, We'd like to expand that to make the service available to all of Stanford Hospital, as well as expand that out to become more of a test that can be performed almost anywhere. With that, we need to figure out a way to scale it appropriately. And on top of that, when it comes to scaling, we want to find ways that we can make this type of testing cheaper as well. So right now, we're sitting at a cost of around $5,000 per test. And while this may seem like nothing in comparison to that $3 billion price point of the first human genome that was sequenced, it is still quite expensive. And getting insurance companies to cover this, we're not at that point. So we want to figure out ways that we can make it cheaper through scaling it appropriately. With that said, one more thing about the cost. If we think about a one-night stay in the ICU, the minimum cost of that is going to be about $10,000 in the United States. So if we're able to reduce the hospitalization time by even a single day through this type of genetic testing, then we should be able to recover that cost.
Great, thank you. Next question. What kind of nanopore sequencing did you use? I have used MinION sequencing before for sequencing T. cruzi and used the de novo assembler Miniasm Minimap. We found these tools were much faster than Canu assembler. What de
Yeah,
novo assemblers have you used?
that's a great question. So the MinION is a smaller, well, it's not necessarily smaller, but it doesn't have as many nanopores embedded into that biofilm. So we were using the Promethean, which has up to 12,000 nanopores embedded on each biofilm. So we're able to sequence at a much higher rate. With that said, the technology is the same. It's just a different flow cell. So using this for something like T. cruzi is fantastic in terms of using the MinION and using these assemblers. It seems like a great approach. Now, we didn't do de novo assembly of our genome. This is something that we certainly will consider, but in order to make variant calls, we aligned to a reference genome, and from there we called the variants. So we didn't do a de novo assembly of each genome. With that said, as you've mentioned, the MinIASM and Minimap is much quicker than Canu, and I think that there are a few more programs that have recently come out that are even quicker than those. So I think that that's something that we certainly will explore moving forward. With that said, we're pretty confident that the variant calls that we make simply by aligning to a reference genome alone are good enough for our application.
Great, thank you. Next question. Do you think you can make this go even faster?
That's another great question. Yeah, I certainly think that we can make this go faster. As technology continues to develop, we're able to see computers that are even more powerful, and with that, I think that we can reduce this turnaround time by a few hours. Yeah.
Great. Next question. Can this be applied to cancer diagnostics?
Also, yeah, another great question. Certainly, this can be applied to cancer diagnostics. The thing is, we would need to probably start thinking about different biological tissues that we're extracting DNA from. So, if we're thinking about a primary tumor or a metastatic tumor, we need to sample that tumor itself in order to get the DNA that's going to be most relevant to these types of diagnostics. So we certainly could apply this technology to cancer diagnostics and make a really quick diagnosis in terms of mutations that may be causative of the cell proliferation, which causes the cancer itself. The only limiting factor there would be if we're talking about solid tumors, cellular tumors, um, then the extraction of DNA would probably take a little bit longer. So rather than taking an hour or so, we'd have to find a more appropriate way to digest that tissue in order to get the DNA out. So that may add a little bit of time to it. But I think we certainly could continue to apply the rest of that technology and come to a diagnosis really pretty quickly for cancer.
Great, thank you. Next question. What patient populations do you think can benefit the most from this technology?
Yeah, so I think when we think about this technology and we think about the speed of the technology, when it comes to critically ill patients, these are the patients that need a really fast diagnosis. So patients that may not survive a day or two in the ICU. We need to make those diagnoses quickly in these patients, and I think that this is where the technology will benefit that type of patient population the most. With that said, I mean, like I showed in this presentation, there were a lot of different types of clinical presentations of these patients that range from neurologic disorders to cardiovascular disorders. And we've not really explored other body systems, but we could think about baby patients, especially with types of kidney disease, where they could definitely benefit from this type of quick sequencing as well. So I think that we're looking to expand, but really keep a focus on the patients who need it most, and those being the critically ill patients.
Thank you. Next question. Do you envision a world where everyone will have their genome sequenced?
Ooh, yes. Yes, I do. I envision a world where everyone who wants to have their genome sequenced will be able to have their genome sequenced. I think based on the developing technology as well as the reduction in cost, it will become available to those who want it. Now, with that said, we can learn a lot about our genome or we can learn a lot about ourselves from our genome, but there are some privacy issues that need to be addressed, and I think bioethicists need to really start addressing these and start thinking about how we can move into creating policy behind genome data and whose data it actually is before we start thinking about sequencing everyone's genome.
Interesting. Thank you. Next question. Do you think it would work with WGA products? Thinking, for example, to single cell applications.
Ooh, single cell applications. Yes, this is always a fun topic to talk about. Right now, nanopore sequencing is moving towards single cell sequencing, and yeah, there are obviously many benefits to single cell sequencing, both at a DNA and an RNA level. And I think we're really just waiting at this point for the technology to be good enough for us to do this on the sequencing platform or the nanopore sequencing platform. And I expect within the next year we'll see methods of this. With that said, we have other sequencing platforms like 10x Genomics that can do single cell sequencing. That's quite good. But in terms of a long read sequencing platform, I think Nanopore is going to come out with it very, very shortly.
Great. Next question. Do you think Nanopore could be used for circulating tumor cells, so using whole genome amplification protocols on single cells?
Yeah, so that's sort of similar to the previous question in terms of single cell sequencing. In our lab, we have tried to do genome amplification from single cells in order to sequence single cells. It didn't work really that great, to be honest. This was a few years ago, though, so the technology wasn't at the point that it is now. But I think rather than using whole genome amplification. I think that simply sequencing native strands of DNA or RNA from a single cell is most likely going to be the way that the technology moves forward. And as the nanopore sequencing technology continues to move forward, we see an error rate in terms of base calling reducing significantly. And hopefully with that reduction in error rate, sequencing single cells will be something that this technology can accomplish.
Great, thank you. Next question. Since timing of testing cannot be predicted, are genetic counselors on call to conduct the manual analysis?
Yes. So, timing of these samples was something that we really struggled with. The case that I presented with you, the patient was airlifted from a different state to the San Francisco Bay Area, and he arrived around midnight. With that, we collected blood and started sequencing around 2 a.m. in the morning. So I was in the lab processing all of this, and it wasn't until early the next morning that we had the genetic counselors start to do their curation process. With that said, yeah, I mean, there were times when we did have to ask our genetic counselors to be up in the middle of the night, which was a big ask. Um, but again, moving forward in terms of our second phase, finding a way that we can appropriately scale this and have enough staff available to have them on hand when needed is something that we are exploring.
Thank you. And we will wrap up with this final question. Are there specific standards used in short reading sequencing that assures fidelity of the sequences compared to the reference sequences?
Yeah, so I think this isn't necessarily a question for short read sequencing, but we can talk about it in terms of any type of sequencing. There's always going to be quality metrics that are used in order to ensure that the sequence that is being aligned to the reference genome is in fact appropriate to be used. And there are many different types of quality metrics. I think in short read sequencing, although I'm not as familiar with it's called a Phred score, a PHRED score, so a Phred score. With nanopore sequencing, it's simply called a quality score. And with that, we set a threshold as to what is an appropriate quality to align. Similar to that of short read sequencing.
Well, thank you again, Dr. Gorzynski. Do you have any final comments for our audience?
No, I think that's it. I really appreciate you taking your time to listen to the developments that we've made in the lab, and I'm excited to be living in 2022 where the world of genomics is booming, and we can see a lot of benefit for human healthcare in terms of these advancements. So with that, I'm excited, and I hope that I've inspired or excited you as well.
Great. Well, thank you again, Dr. Gorzynski, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 21st of 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.